So we're going to move now into the scientific part of the conference, and I'm really very, very pleased to welcome Dr. Peter Crino as our next speaker. Pete, as he's like to be called, is a internationally recognized physician scientist in the area of developmental brain disorders. Tuberous sclerosis is one of his areas, but certainly not the only one. And Dr. Crino received his MD from Yale and then his Ph.D. from Boston College. He went on for training, moved to the Philadelphia area at University of Pennsylvania, joined the faculty at Temple, and recently he's just become chairman of neurology at the University of Maryland. I know Pete through mostly through study section activity in Washington, which has been a pleasure to get to know him, and really look forward to hearing about mTOR and its relationship to this condition.
Great, thank you very much. It's really a pleasure to be here. I want to thank the organizers for inviting me. Obviously, Steve, it's a real pleasure. I do want to indeed give a shout out to Dr. Malbari, because the last time I was here lecturing, you were just getting this process off the ground. We had some conversations, and indeed, you've done an outstanding job to get everybody together, so you're to be credited. It's a true honor for me to give a lecture in front of Dr. Rapin in this incredible symposium. I read your Brain Dysfunction in Children book when I was a grad student, and it really helped to shape a lot of my interest in this field, so it's truly an honor to be here. So I'm going to talk to you a little bit today about the mTOR cascade. These are just some disclosures, nothing felonious. And the mTOR cascade has basically exploded in terms of our understanding of neurodevelopmental disorders, but actually also getting into senescence and dementing illnesses as well over the past 5 years. And so the talk really this morning, I'm going to try to go quickly. I have a lot to cover, so you can give you sort of the full spectrum of understanding what the mammalian target, or now as it's called, the mechanistic target of rapamycin cascade is all about, and we'll give you some context for understanding tuberous sclerosis. So my talk is really going to be about perspective. So this is a slide that's showing a picture of Machu Picchu, but this is really what everyone thinks of as Machu Picchu. So it really depends on where you're standing. And the same thing when we think about malformations of cortical development and disorders that are linked to the mTOR cascade, there are perspectives that involve whole brain regions, whole brain areas, as well as down to single neurons and subcellular domains in the brain. And you'll see that this has been quite relevant to understanding this process. So the tuberous sclerosis complex genes encode these two proteins, TSC1, TSC2. There's a third component of the complex known as TBC1D7, and you'll be hearing a lot more about this from Mike and from Elizabeth. But this was the sort of the overall view of the mTOR signaling cascade, and it's really been implicated in a number of broad areas. So cancer, metabolism, and most recently and relevant to this conference is in brain development. This sort of remains the keystone or the platform, if you will, for sort of trying to understand this. But the level of complexity in terms of understanding neurological disease has really exploded. This pathway here, when it's active and when it's functioning, is largely devoted to enhancing the processes of cellular growth and cellular proliferation. When it is shut off, though, this is something that's growing getting more attention. It's also linked to a process known as autophagy, which is a position of cellular conservancy where the cell learns how to sort of conserve its resources under settings of stress. And indeed, the mTOR cascade has been implicated now in Alzheimer's disease and Huntington's disease and in Parkinson's disease. Because of the limits of time, I can't go into these. I wrote a review on this recently if you'd like to learn a bit more. But suffice it to say, it's really an exploding field. Today I'm going to speak to you about malformations of cortical development, of which tuberous sclerosis is one subtype. And this will include a number of disorders, so TSC, focal cortical dysplasia, specifically type 2 focal cortical dysplasias, the complex of hemimegalencephaly, one massively enlarged and dysmorphic hemisphere, or megalencephaly, when both sides of the brain are enlarged, a novel disorder called Pretzel syndrome, which I'll describe in brief, and ganglioglioma, which I won't talk about too much. As I don't have to tell this audience that these disorders are highly associated with medically intractable epilepsy, may be variably associated with intellectual disability and with autism spectrum disorder. In these disorders, the malformations that we see are typically focal and can be detected on brain imaging, such as MRI, but not always. There is indeed frequently co-localization between the MRI findings and the EEG findings, such as spikes or ictal onset zone in epilepsy patients. But it is a true statement that there is often patients who are found histopathologically to have malformations that have radiographically normal studies. And many cases of so-called non-lesional neocortical epilepsy actually have turned out to be be dysplasias. Now, the process of forming malformations de facto as malformations of cortical development reflects changes during embryogenesis. We know that the neocortex forms during weeks 8 to 20 of human gestation. All of these malformations are characterized by a series of structural or morphological abnormalities, so changes in cortical lamination, changes in the individual cellular morphology, changes in gyral structure. The patterns of inheritance of many of these disorders have only been recently understood, let's say the past 5 years. These can be autosomal disorders, such as tuberous sclerosis, for example, which can be dominant or recessive disorders, and these are inherited. In this picture, pretty much every cell in the body has inherited a mutation in a candidate gene. Conversely, a number of these disorders are so-called sporadic disorders, and they result from somatic mutations that happen in neuroglial progenitor cells, and therefore the mutational effect is consigned only to the central nervous system, not the rest of the body, and I'll give you some examples of this later. Now, in some cases, I do want to point out that germline mutations, as in TSC, can happen where it's a de novo mutation. When sperm and egg come together, there's a mutation. So it's not inherited, but it's nonetheless a germline process. So really, we have inherited spontaneous sporadic germline and spontaneous sporadic somatic mutations. Now, malformations of cortical development are neurodevelopmental disorders. And I say that to remind you, so in this two pictures, here's a MRI scan showing two dizygotic twins. This twin does not have TSC. This twin does have TSC. Here you can see a small nodule in the brain already present there. And this is a fetal MRI depicting hemimegalencephaly. The reason I show you this is to say that these are processes that are happening during in utero development. There's a process to this. There's a period of time. And it does begin to sort of tickle our fancy about the possibilities for critical periods of intervention during embryogenesis. But it's important to realize these don't start all of a sudden. These have a genesis. Now, we know that the disorders that I'm going to talk about today, so-called mTORopathies, are disorders that are linked to aberrant or increased signaling through the mammalian or mechanistic target of rapamycin cascade. These are due to gene mutations in mTOR regulatory genes, either loss of function mutations in inhibitors or gain of function activating mutations in activating proteins. And we know that the mutation has an effect at each step in cortical development. So during the proliferative phases, it may have an effect on cell proliferation. On the migratory phases, it may affect how the cell physically moves from the ventricular zone out to the nascent cortical plate, how the cells begin to actually recognize their laminar destiny, and then finally in the process of cortical organization, when neurons in the neocortex have achieved the laminar destination, begin to extend dendrites, axons, and make functional synaptic connections. The effect of the mutation can be seen to sort of echo through each one of these steps of cortical development. The general model that's been accepted, I would say over the past decade or so, is a model that looks something like this. And it really can be applied to lesions that are either small, medium, hemispheric, or even bihemispheric. And that is, in the ventricular zone, there is a mutation that affects either a single progenitor cell or a number of progenitor cells. There's ongoing cellular proliferation. The cells migrate up to cortex, typically abnormally. And what we have is sort of a gradient of severity between cells where there's regions where there's lots and lots of mutations, areas where it's a modest level of somatic mosaicism. So in this case, the blue are normal cells genomically. The red cells have a mutation, in a case such as a somatic mutation. Or malformations where there's very few numbers of mutated cells. So if you look at a lesion such as this is a focal cortical dysplasia. This is a grossly disorganized gyrus. What you can see is that if you look at the cellular features, there's changes in cellular morphology. There's a characteristic feature of many of these malformations, which is cytomegaly or cellular enlargement. The cells are dysmorphic in shape. There are the presence of so-called cytomegalic dysmorphic neurons, which I'll show you, and a very unique cell type, which is found in malformations such as type 2 dysplasia, tubers and tuberous sclerosis, and hemimegalencephaly, which are called balloon cells. So here's what the normal lamination of neocortex looks like, and by comparison, this is what an area of focal cortical dysplasia type 2B looks like, a complete disorganization of laminar architecture in the presence of these very, very large cells here, which are called balloon cells or dysmorphic neurons. These are what balloon cells look like, these ovoid eosinophilic cells with laterally displaced nuclei that are very, very large. And this is what a cytomegalic dysmorphic neuron looks like. Very, very aberrant morphology, often very twisted apical dendrite, multiple basal dendrites, and just a very, very abnormal cell type. So if you take that histopathology and superimpose it on this model, you get a better sense of what happens in, for example, a somatic mutational event. The mutation occurs, cells undergo mitosis, they migrate up. The cells in red contain the mutation, the cells in tan do not have a mutation, and what you end up with is a focal malformation with some dysmorphic cells, some, for example, balloon cells, and then you have other adjacent cells scattered within the lesion itself. Now, what's interesting is there's a huge amount of heterogeneity in these lesions. As we know, there can be lots and lots of dysmorphic neurons, lots and lots of balloon cells, or very, very low numbers. In addition, something that's interesting is if you look in the surrounding cortex, and this is particularly relevant to tuberous sclerosis, there may also be cells that contain mutations scattered out here. And so the actual extent of the lesion is sometimes very, very difficult to define. So this was the mTOR signaling cascade as of 2001. We knew that there was a link between growth factor signaling through a number of canonical enzymes that are kinases signaling through, and that somehow this molecule here, mammalian target of rapamycin, had a downstream effect largely on protein synthesis to control protein translation and cellular growth. The function of mTOR in the brain, if you look back, let's say even into the 1990s, was largely unknown and largely underappreciated. The TSC1 and TSC2 genes were cloned and identified respectively in 93 and 97. And once that happened, the platform exploded. And over the past 10 years, we've now filled in the cascade with numerous other regulatory proteins, all encoded by regulatory genes. We now know that mTOR plays an important role in all of these different cellular functions in the brain. All right, the general model for tuberous sclerosis as a platform disorder, as a paradigm disorder, is that there's a loss of function mutation in TSC1 or TSC2, which means that it can no longer do its job, which is to constitutively inhibit mTOR. When that happens, mTOR is constitutively activated. There's no way to stop mTOR activation, and as a consequence, you have growth of so-called hamartomas, such as cortical tubers, for example. And this general model is a good way to sort of think about mTORopathies or disorders of mTOR signaling. Now, the first paper to show that mTOR was activated in the CNS in tubers and cortical dysplasia was published in my lab in 2004. And the model essentially was this. If you look within a cortical tuber, here's the signaling cascade. In the setting of loss of function mutations of TSC1 and TSC2, you see increased signaling through mTOR. The way we know this is to look at so-called biomarkers, in this case, phosphorylation of this protein, S6, or phosphorylation of this protein, 4-EPP1. These are very good biomarkers for mTOR activation in tissue or in blood. If you look in control cortex, you can see that at baseline levels, there's very low levels of mTOR signaling. So there's really now three broad vectors that we have been able to identify that are linked to malformations of cortical development. The first is the group here, which is really the growth factor effect, starting with insulin-like growth factor, but other growth factors, signaling through to TSC1 or TSC2, then signaling through a small protein known as Reb to activate mTOR. The second is an amino acid signaling cascade, which leads to a complex known as GATOR1 and two particular proteins, DEPTEC5 and MPRL3, which serve to regulate mTOR activity in the setting of nutrient deprivation versus nutrient repletion. The third is an energy-dependent cascade, which senses ambient levels of ATP and AMP in the cell and drives mTOR activation when energy is adequate. The general model here is that mTOR signals to enhance cell growth when there are adequate levels of growth factors, adequate levels of nutrients, and adequate levels of energy in the cell. Mutations in a number of these genes have now been identified in a number of malformation syndromes. All of them confer a condition of hyperactivity of the mTOR signaling cascade. And so this whole panoply of so-called mTORopathies, whether it's hemimeg or cortical dysplasia, or here the paradigm disorder tuberous sclerosis, the central platform here is that the mTOR cascade is activated. Okay. What are some of the disorders and where are the genes? We know there's a disorder known as Pretzel syndrome, which is found in the old order Mennonite community in Lancaster, which has megalencephaly as its phenotype. Mutations in the GATOR1 complex have been associated with cortical dysplasia and hemimeg. Mutations in PI3 kinase, AKT, or TBC1D7 have been variably associated with hemimeg, megalencephaly, cortical dysplasia. Recently, activating mutations in mTOR have been associated with focal cortical dysplasia. And we know that there's a number of different inheritance patterns, as I mentioned, either germline or sporadic, that give rise to these mutations. Here's rapamycin or sirolimus, which is similar to the drug everolimus, and so we have a very, very druggable target right here with a very good inhibitor of this. So it's not surprising that we've moved on rapidly to drug trials in the past few years. So this whole picture gives you kind of the geography currently of focal malformations of cortical development that have all been linked to the mTOR signaling cascade. Because of that, a few years ago, my lab coined the term mTORopathy, that these are disorders of mTOR signaling. So I showed you before how this would work for TSC, but actually now we know that for activating mutations in PI3 kinase, AKT, and mTOR, or disinhibiting loss of function mutations in PTEN, DEPTIC5, or MPR3, the net effect is to enhanced mTOR signaling. So what is mTOR? Well, we know that mTOR is a serine threonine kinase that functions in a number of different cellular processes. The take-home is that its main cellular function is to regulate protein synthesis and to enhance cellular growth and proliferation. There's two isoforms, mTORC1, which is defined by its binding to this protein Raptor, and mTORC2, which is defined by its binding to Richter. My comments today will be confined only to mTORC1. The reason this is relevant is that mTORC1 is specifically sensitive to the mTOR inhibitors rapamycin, sirolimus, or everolimus. Here's mTOR. It's actually part of a very, very large protein translational initiation complex. This is messenger RNA. And so really the net effect of mTOR signaling is to drive and to facilitate protein synthesis via modulation of protein translation deep within this translational machinery. This is rapamycin, also known as sirolimus. It's a multi-ring structure that is found endogenously in a soil bacteria on Easter Island. There's been synthetic analogs such as everolimus, which have been made in the laboratory just by a simple modification of this to a carboxyl group. It is an immunosuppressant drug, but it also has effects, obviously, in other aspects, especially in the central nervous system, on brain function. The way the drug's supposed to work is that it binds to a protein known as FKBP12. This complex then comes in and sits on mTOR. It's not a direct kinase inhibitor, but what it seems to do is pinch off mTOR's ability to recognize its substrates. So, if malformations of cortical development are neurodevelopmental disorders, then mTORopathies are neurodevelopmental disorders. Therefore, we can begin to think about when are critical periods for intervention to either prevent the formation of malformations in the brain, or at least to begin to prevent some of the clinical phenotypes such as epilepsy, autism, and intellectual disability. We know that the mTOR cascade is turned on during fetal development. So this is a study from my lab looking at fetopsy specimens, and as early as 23 weeks gestation in a TSC fetus, you can see that the mTOR signaling cascade is activated. So think about it. If we're going to use clinical mTOR inhibitors in a 3-year-old, that cascade has been activated for all of 3 years, plus a fair number of weeks during gestation. So this is a process that starts pretty early on. So let me tell you a little bit about each of the 3 vectors. So the growth factor vector, which is the one that canonically signals from growth factors all the way through to TSC1, TSC2, and then to mTOR, accounts for a number of malformations. So in addition to tuberous sclerosis, which gives rise to a focal cortical dysplasia that we call cortical tubers, there's also mutations in PI3 kinase, PTEN, which regulates PI3 kinase, AKT, and mTOR, all of which give rise to focal malformations of cortical development. Some result as germline effects, some are somatic effects. All of them have the same histopathological features. Tuberous sclerosis, obviously, is the purpose of this conference today, is a multi-system disorder that affects the skin. Here's a renal lesion, here's lung lesions here. But really, its most devastating effects when we think about sort of functional impairments in patients are epilepsy, intellectual disability, and autism. Again, if you look at a cortical tuber in a gross autopsy specimen here, it's a focal area within the cortex. And here you can see the mutation gives rise to cells that are all phospho-S6 positive due to activation of the mTOR cascade. We know that if you knock down TSC2 or TSC1, if you knock down PTEN, or you activate mTOR, PI3 kinase, or AKT, the phenomenon on cells in the culture dish are exactly the same. You have an massive increase in cell size, which is similar to what we see histopathologically in these lesions, and that if you give rapamycin to these cells, you can immediately reverse the effects. So the effects on cell size are highly plastic. A lot of work has been done in mouse models as well. Some of the work that Mike Wong is going to talk about today also has demonstrated this in the intact animal in vivo. If you think about focal cortical dysplasia, you can see here, defined radiographically, here's phospho-S6, here's the histopathology. Focal cortical dysplasia has most recently been found to be due to mutations in mTOR itself. These are activating mutations in the various domains along mTOR. And indeed, if you look at the profiles of phospho-S6 labeling in FCD2A or 2B, it's virtually identical to what you see. So they're effectively endophenotypes of one another, even though the mutation here is TSC1 or 2, and here the mutation is in mTOR. Hemimegalencephaly, massive enlargement of one hemisphere, has robust mTOR activation as evidenced by phospho-S6 levels. So here's the enlarged hemisphere, here's the control hemisphere. You can see clear differences in S6 immunoreactivity. There's been a number of studies published from a variety of different groups. I can't even name them. There's so many of them around the world, but certainly Bill Dobbins' group, Stephanie Bolak's group, Ann Podori's group, a number have identified mutations in these mTOR regulatory genes, which you can see along this cascade here, which confer activation of mTOR. We know along some of the genes exact addresses where we can confer differences in phenotype, either hemimegalencephaly or megalencephaly. That's how far the field has actually come in the last 5 years. Focal cortical dysplasia and hemimegalencephaly, from a mechanistic perspective, though, are not the same thing. And it's very important to sort of try to understand this because it's relevant to the phenotypes of these disorders. In cortical dysplasia and hemimeg, the presumption is that there is an isolated focal malformation which contains an admixture of cells. In blue, these are plus cells. These are cells that do not have any mutation. In red, these are cells that express high levels of mTOR activation and contain a heterozygous mutation. So you have a regional heterogeneity, and then within the lesion, you have a lesional heterogeneity. In contrast, in tuberous sclerosis, the background of the brain is a condition of heterozygosity, plus minus. There are no normal cells in the TSC brain. All of them are plus minus, upon which is superimposed a group of so-called minus cells. These are cells that lack TSC1 or TSC2 fully. And so here you have a whole sea of normal brain upon which is superimposed a lesion, whereas here you have a sea of maybe normal brain upon which is superimposed a lesion. The degree of allelic variance here, the number of mutated cells in FCD and hemimeg has been calculated. It ranges from about 1 to 30%. That means anywhere from 1 in 100 to 30 cells per 100 can have an actual mutation. In TSC, the allelic variance 100%. Every cell is a heterozygote, and many of the cells contain somatic allelic variance, giving rise to a greater 15% load. So when you look at this lesion as an FCD, or you look at this lesion as a tuber, whoops, I don't know what that was. It's very, very different proposition. The other difference, of course, in tuberous sclerosis is that the lesions tend to be multiple. So not only do you have multiple tubers, you also have areas of so-called perituberal cortex, which have been shown to contain tissue abnormalities. So the tissue is not wholly normal. Moreover, there's been some recent studies to suggest that subcortical areas, such as the basal ganglia and the thalamus, may also be abnormal in TSC. If you look at autopsy specimens of TSC, what you can see is that here's a tuber right here. This is labeling with glial fibrillary acidic protein, GFAP. It's a marker of reactive astrocytosis. Here's a tuber here. Here's a tuber here. But if you look scattered throughout the rest of the brain, even in areas where it's not really a tuber, you see things like radial migration lines. You see small little isolated areas. And indeed, if you look histopathologically, the TSC brain is just sort of peppered with many, many small areas of tissue abnormality, so-called micropathology. This is not a feature of focal cortical dysplasia. So this is a very different feature of TSC compared to FCD. And of course, this belies the clinical phenotypic difference between FCD, which is largely an epilepsy phenotype, and tuberous sclerosis, which thankfully I put autism on my slide, which has a much more rich intellectual and behavioral phenotype than FCD. And of course, as I mentioned, there are often multiple cortical tubers and even subcortical tubers. So, number of different gene mutations along the canonical growth factor side, which give rise to FCD, hemimeg, or MEG, all have a high association with epilepsy, and they raise the possibility for clinical trials. And I think Elizabeth or Mike are going to talk about the data from EXIST-3, which is using everolimus to treat seizures. All right, the amino acid vector. This story has become incredibly exciting and incredibly complicated over the last couple of years in that we found that there are mutations that are part of this complex GATOR1 in DEPDEC5 and NPRL3. These confer a phenotype of focal cortical dysplasia and hemimegalencephaly. NPRL3 confers a phenotype of focal cortical dysplasia. Again, some of them are germline, some of them are somatic. They have the same histopathological features as what we see in FCD and hemimeg. This is an example of so-called bottom of the sulcus dysplasia. It's a dysplasia that's sitting right here. These are particularly vexing as part of pre-surgical evaluations because they're often tough to evaluate and tough to pick up by EEG. If you look histopathologically at these, they look just like FCD type 2B with large balloon cells that are phospho-S6 immunoreactive. This complex here, Gator1, which is comprised of deptic-5 and NPRL3, is actually a complex that regulates mTOR signaling in the setting of amino acid deprivation. If there's not enough amino acids in the cell, this complex turns off mTOR. It tells mTOR, don't signal, don't facilitate growth, there's not enough nutrients. In the setting of loss of function mutations in these, mTOR is constitutively activated, irrespective of what's going on the nutritional aspect of the cell. By the way, we now know there's a Gator2, and there's a cestrin and a castor, so the story gets even more and more complicated. This is a study that we published a little while ago in the Annals looking at a family of 2 boys who had a deptic-5 nonsense mutation. They both had focal cortical dysplasia and epilepsy. Interestingly, others in the family had features that were unclassified epilepsy, febrile seizures, or focal epilepsy, but no cortical dysplasia. And here's a very interesting picture of these 2 brothers. So here's the 2 brothers here. They have the same mutation. And what you can see here, here's the one brother who basically has a hemispheric dysplasia. And here's the other brother who has a small focal cortical dysplasia. So this phenomenon, different phenotype in the setting of common genotype, is known as pleiotropy. And an explanation for this we don't have for many of these disorders. Tuberous sclerosis is a disorder characterized by extreme heterogeneity in terms of the clinical presentation and extreme pleiotropy. With any one patient's mutation, we really cannot predict what the landscape of what their clinical progress is going to be. But nonetheless, if you look at resected specimens in both of these, you can see a characteristic dysplasia phenotype with hyperactivation of the mTOR signaling cascade. Mutations in deptic-5 also give rise to hemimegalencephaly as well as megalencephaly. This is a mutation in NPRL3, which is the binding protein of deptic-5, which confers hyperactivation. This is a paper also in the Annals, which confers hyperactivation of the mTOR signaling cascade. In vitro, work in my lab, we've worked to use shRNA to knock down these genes. If you knock down deptic-5 or NPRL3 compared to control neurons, you get a marked enlargement in cell size, here depicted graphically. So here's deptic-5, here's NPRL3. Interestingly, NPRL2, which is a binding protein, knockdown does not seem to affect size. We don't have an explanation for that. And if you treat these cells in a dish with rapamycin, you can see a very robust effect depicted here in reducing the cells back to a normal size, suggesting that rapamycin, aka mTOR inhibitors, can affect cell structure here. So deptic-5 and NPRL3 confer some very, very interesting focal cortical dysplasia or hemimegalencephaly phenotypes. There has been discussion about using mTOR inhibitors to treat seizures in these disorders, but it has not come to the fore just yet, but it's certainly being discussed at the meetings. Last is the energy vector here, which is signaling through the AMPK pathway, and in particular mutations in this protein or this gene right here, STRAD. This gives rise to a disorder that I mentioned is seen in the Old Order Amish community in Lancaster, Pennsylvania. It's a very interesting phenotype. It's incredibly rare. I'm not suggesting it's a burgeoning public health problem, but it is a rare disorder that confers a very interesting insight into this cascade. The idea is in these children, they have a macrocephaly phenotype with megalencephaly. They have a big brain and a big head. They have severe epilepsy with 100% penetrance. They have profound neurocognitive deficit. They have subependymal collections of cells and focal cortical dysplasia. So even so the gene mutation is way out here, in some ways it looks radiographically a little bit like tuberous sclerosis, which is kind of interesting. As I mentioned, there's a 100% penetrant phenotype. Going back, we've been able to identify cases as far back as the 1740s, so this has been around for a long time. It's a founder mutation in this gene. The gene's called STRATA right here, and the idea is that if you lose function of that, you lose the ability to regulate mTOR, and you turn on mTOR signaling, similar to all the other disorders I've shown you previously. We had a recent identification of a non-founder case in a consanguineous Indian pedigree, suggesting that this mutation is probably out there in the world as well. If you look histopathologically, the neocortex architecture is actually reasonably intact, but there are scabillions of heterotopic neurons down in the subcortical white matter, right in through here. If you look at S6 signaling in fibroblasts and in blood from patients, here's a control, me. This is heterozygous parents of these kids, and then this is the actual kids here. The other name for pretzel syndrome is polyhydramnios megalencephaly symptomatic epilepsy syndrome, which I think is really dopey, so I don't use that. But you can see even in fibroblasts, there's high levels of S6 activation. If you do migration assays using a scratch test with neural progenitor cells, if you take normal cells, they're able to migrate just fine to close a scratch. If you knock down STRAD, they completely lose their way. If you give them rapamycin, you can completely reconcile and rescue that phenotype. So it's wholly mTOR dependent. If you use uterine electroporation to actually knock down STRAD, so here you externalize the uteri, inject an shRNA, then use electroporation to introduce the gene, you can see GFP immunofluorescence here where we make sort of a microdysplasia. I won't belabor the point, but here's control and here's the knockdown. All of these green cells are supposed to be up here. All of these green cells are supposed to be up here. So you can induce an experimental focal cortical dysplasia. If you give rapamycin, you can completely rescue this lamination defect. We've used, in collaboration with Jack Parent, we've taken fibroblasts to make induced pluripotent stem cells and then to convert these and derive these into iPSC-derived neurons. These are what they look like with tau staining. These are the iPSCs here. There's no STRAD in any of the proband cases. There's STRAD in the controls. There's hyperactivation of S6 in the probands, but not in the controls. The cells are larger, as we would expect. They have abnormal polarity. So here what we see in normal neurons, we see MAP2 in the dendrites and tau in the axons, but in these cells, MAP2 and tau are overlaid. So they have a complete loss of cellular polarity. They express high levels of phospho S6, and if you patch clamp these cells independently and spontaneously spike. So based on all of this data, what we actually did was we picked a cohort of patients, and we didn't go to the adult patients. We actually went to younger kids prior to the onset of epilepsy. So at 4 and 5 months of age, we gave these children sirolimus. And again, the penetrance of epilepsy in this group is 100%. And what we found was over the year following our treatment, once we had adjusted the doses over time, we had a near cessation, or actually a prevention of any seizures occurring in these children. This was published in Science Translational Medicine. And the biggest finding really was the mortality of this disorder is 38% from status epilepticus. We have had no mortality with rapamycin therapy. This is a 5-year follow-up at this point in time. Again, we've had no mortality and no status epilepticus. So this trial was really the first small targeted trial to show that you can prevent seizures by manipulating the mTOR signaling cascade. So I'd like to think it sort of laid the way a little bit for EXIST-3 in terms of its role in tuberous sclerosis. So what I've showed you is that there are a number of mTOR regulatory genes that confer differential cellular phenotypes. All of these phenotypes are associated with medically intractable epilepsy, some of them associated with intellectual disability or with an autism phenotype. And that mTOR inhibitors clearly in the dish and in the animal and in cell culture can affect mTOR signaling and in some cases can actually affect the clinical outcome. Now, whether these will work for all of these disorders, some of these disorders, or only a select few is very unclear. What are the limitations to mTOR inhibitor therapies? Why don't mTOR inhibitors cure everybody? Well, there's a number of explanations here. Some of it is timing. When do we start the medication? We may have to start very early, and if you think of those images I showed you of critical periods, we may need to be thinking about using these drugs prior to the onset of any neurological symptoms in the patients. Monitoring mTOR inhibitions, are serum drug levels adequate? Are tissue levels adequate? Can patients tolerate the drug? And a big question that's in the literature really now back and forth is when do we stop using these medications? Finally, the last idea that's very complicated is that if you look in these lesions, there's a very, very complex microenvironment there, not the least of which is a very robust inflammatory response. It's unlikely that all of these changes that are happening here are actually due to mTOR activation. Some of them may be autonomous and sort of have taken on a life of their own over time. So just giving mTOR inhibitors will likely not wash away all this stuff. And I'll just skip this here. Sorry. And this is just a list of all the different mutations that have been associated with malformations of cortical development in the mTOR signaling cascade. And I just want to thank my laboratory, my funding, and again, thank you very much for inviting me.
What a tour de force. Thank you, Pete. Questions? Jose, can you shout? Maybe it'll take the mic up.
So tubers have been reported in the cerebellum. I'm not aware of anybody finding a hypothalamic or a basal ganglia tuber. Focal cortical dysplasias, by their nomenclature, are cortical malformations. So no. Now, in hemimegalencephaly, often when the entire hemisphere is enlarged, the basal ganglia and thalamus are also enlarged as well. So it's sort of a global enlargement phenomenon. But the malformation, we like to think, probably derives from neocortical progenitor cells in the VZ. And so that's probably the substrate for this. And certainly in TS, there's no subcortical manifestations other than the rare cortical tuber and a couple of reports that have shown dropout of cerebellar Purkinje cells in TSC.
You know, Pete, as a lysosomal storage disease investigator, I noticed the lysosome looming large at the bottom of many of your slides. You didn't actually mention it, but just point out to students and postdocs in my lab, you know, that mTOR, when it's active, it's sitting on the surface of the lysosome. Right. But, you know, in relation to that, these balloon cells that you mentioned with displaced nuclei to the side of the cell, what's in the cytoplasm of those cells that's causing that?
Yeah, no, so it's interesting. There's been a number of studies. There's lipids. There's a lot of proteins, a lot of ribosomes. There's a paper that was published. Mustafa Sahin's lab looked and showed that there were inclusions that were P62 sequestrosome ubiquitin positive. So maybe even though the cells have hyperactivated mTOR, they may actually be sort of weirdly activating the autophagy cascade as well. If you do Saddam black, it's lipid. So it's an admixture of things in the cell. We sort of wondered, what is the stuff that makes the cell big? Yeah. I don't think it's water. I think it's stuff.
Yeah. Wow. Interesting.
Cellular stuff.
Other questions? Well, Pete, thank you so very much for that.